Hello everyone, I am Satish Kumar Pandey. I am going to give my presentation on the topic vaccine and immunization. As you know, for the last 1.5 year, everybody is talking about immunity, immunology, vaccine, and vaccination. They are more familiar about these biological but so I thought I could present my seminar on this topic which everybody can understand the topic easily. In this presentation, I am going to talk about basic thing about vaccine. So we come to the second slide. What is a vaccine? Vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. In other words, a vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed form of the microbe. What is immunization? Immunization is the process of giving a vaccine to a person to protect them against disease. Now we move to the next slide. In the scientific literature, it is documented the practice of immunization was adopted 100 years back. Buddhist monk drank snake venom to confer immunity to snake bite, and smearing of skin tear with cowpox to confer immunity to smallpox was practiced in 17th century in China. That clearly indicate that immunization is an old process. The term vaccine and vaccination are derived from a Latin word variola vaccine. Edward Jenner is considered the founder of vaccinology in the West in 1976 after he inoculated a 13-year-old boy with vaccine virus, that is cowpox, and demonstrated immunity to smallpox. Moreover, in 1970, the first smallpox vaccine was developed. Over the 18th and 20th century, systemic implementation of mass smallpox immunization culminated its global eradication in the year 1970. Another scientist, Louis Pasteur, and his experiment is furthered the development of live attenuated cholera vaccine and inactivated anthrax vaccine in human. Similarly, plague vaccine was also invented in the late 19th century. Between 1890 and 1950, bacterial vaccine development proliferate, including BCG vaccination, which is still in use today. And on the other hand, viral tissue culture method developed from 1950 to 1985 and led to the advent of Salk inactivated polio vaccine, and after that live attenuated oral polio vaccine. It is also worthwhile to mention here mass polio immunization has now eradicated the disease from many regions around the world. On the basis of vaccine preparation, it is broadly divided into the following type: live attenuated vaccine. An attenuated vaccine is a vaccine created by reducing the virulence of a pathogen but is still keeping it viable. Moreover, attenuation takes an infectious agent and alter it so that it becomes harmless or less virulent. On the other hand, inactivated vaccine is a vaccine consists of virus, particle, bacteria, or other pathogens that have been grown in culture and then killed to destroy disease producing capacity. In subunit vaccine is a vaccine that contains purified part of pathogen that are antigenic or necessary to elicit a protective immune response. Toxoid vaccine is an inactivated toxin whose toxicity has been suppressed either by chemical or heat treatment, while other properties typically in like immunogenicity are maintained. In conjugated vaccine is a type of vaccine which combine a weak antigen with a strong antigen as a carrier so that immune system has a strong response to the weaker antigen. DNA vaccine A DNA vaccine is a type of vaccine that transfects a specific antigen coding DNA sequence onto the cell of an immunized species. DNA vaccine work by injecting genetically engineered plasmid containing the DNA sequence encoding the antigen against which an immune response is shown. So the cell directly produces the antigen and thus causing a protective immunological response. Recombinant vector vaccine. A vector vaccine is a vaccine produced through recombinant DNA technology. This involves inserting the DNA encoding an antigen such as a bacterial surface protein that stimulates an immune response into bacteria or mammalian cell, expressing the antigen in these cells and then purify it from them. Ribonucleic acid vaccine or messenger RNA vaccine is a type of vaccine that use a copy of molecule called messenger RNA to produce an immune response. mRNA vaccine are a new type of vaccine to protect against infectious disease. mRNA vaccine teach our cell how to make a protein or even just a piece of protein that trigger an immune response inside our bodies. Properties of ideal vaccine: it should be free from carcinogenicity and toxicity, able to induce both humoral and cell mediated immunity, should be free from any pyrogenicity and mutagenicity, biocompatible and biodegradable, not cause any non-specific immune effect. This is the pictorial representation showing how attenuated vaccine work. As I said that attenuated vaccine is a vaccine created by reducing the virulence of a pathogen but still keeping it viable. In another word, attenuation takes an infectious agent and alter it so that it becomes harmless or less virulent. Figure A shows the attenuation of virus by a serial passage of the virus through a foreign host. Figure B shows the attenuated bacteria by chemical treatment or subculturing method. Figure C shows the live attenuated vaccine containing function copy of pathogen that have been weakened and recognized by antigen presenting cells. Then T helper cell activate and produce antibody and immune response along with development of memory cell. In this slide you can see how inactivated vaccine work. Here I took Covaxin as an example. How does Covaxin work? As you know that Indian company Bharat Biotech partnered with National Institute of Virology and ICMR Indian Council of Medical Research to develop an inactivated coronavirus vaccine known as a Covax. In the development of Covaxin, the first step is to produce or culture, and often after that used to treat them with a chemical called beta propiolactone. The compound disable the coronavirus by binding to their gene. The inactivated coronavirus could no longer replicate, but their protein, including spike, remain intact. After the inactivation of virus, it is mixed with a tiny amount of aluminum based compound called as an adjuvant. Adjuvant stimulate the immune system to boost its response to a vaccine. In this slide, we discuss the subject of subunit and DNA vaccine. As I mentioned in a earlier slide, subunit vaccine contain fragment of pathogens such as protein or polysaccharide whose combination are selected to induce a strong and effective immune response. As you can see in the diagrammatic figure that with the help of antigen presenting cell, that antigen are recognized by T helper cell as with the real viral infection. Generally, subunit vaccine only induce antibody mediated immunity. Figure B show how the DNA vaccine work. DNA vaccine consists of component or part of infectious agent modified in such a manner that no harm or disease are caused, but ensuring that when the host is confronted with that infectious agent, the immune system can adequately neutralize it before it cause any ill effect. This slide shows that how conjugated vaccine work. In a conjugated vaccine, the weak antigen is covalently attached to a strong antigen, thereby eliciting a strong immunological response to the weak antigen. Most commonly, the weak antigen is a polysaccharide that is attached to a strong protein antigen. You can see that in this slide that a specified virus antigen is integrated with a strong antigen, that is a tetanus toxoid protein, which have ability to induce the immune response and which has the ability to trigger the immune response. In this slide, we talk about the vaccine administration route. Vaccine are educated or administered in following route, that is oral or intramuscular, subcutaneous, intradermal. This slide show how vaccine generally work in the human body. You can see there that a weakened form of virus is injected into the body. The body think big virus is a threat, then immune system start to build up a lot of antibody. Once the pathogen attack the body, the antibody are ready to catch them and destroy them. That is how the vaccine work. In this slide, we can see the delivery system that is used to promote uptake of antigen in vaccine. Steps in vaccine development: preclinical study, that is a vaccine tested in animal study for efficacy and safety. After that, phase 1 clinical trial, some small group of healthy adult volunteers received the vaccine to test for safety. After first clinical trial, second phase clinical trial will be conducted and vaccine is given to people who have characteristics similar to those for whom the new vaccine is intended. In clinical phase third, vaccine is given to thousands of people and tested for efficacy and safety. In phase fourth, post marketing virulence, ongoing study after the vaccine is approved and licensed. In after the fourth step, human challenge study in which a vaccine is given followed by pathogen against which vaccine is designed to protect. Such trials are uncommon in people as they present considerable ethical challenge. Thank you very much.